Hello everyone and welcome back to sqadia.com. So the topic of discussion today is gluconeogenesis. So what is gluconeogenesis? Let us divide the word into three parts, as you can see in the writing here. Gluco means glucose, neo means new, and genesis means generation. So if we look at the whole word, it would be generation of new glucose. Gluconeogenesis is the production of glucose from non-carbohydrate molecules. And what are these non-carbohydrate sources? They can be lactic acid, amino acids, glycerol, and some of the odd-chain fatty acids. The substrate molecules are derived from breakdown of lipids, carbohydrates, and protein. We will discuss all of them in a bit. Now the next question is that where does it occur? It mainly occurs in hepatocytes and some of it occurs in the kidneys. Most gluconeogenesis reactions occur in the cytoplasm, but two reactions occur in mitochondria. Now it can be required for two main reasons. One is that whenever there is low blood glucose in the body. This condition is commonly called hypoglycemia. Here body requires to produce more glucose. And the other reason is brain. Why brain? Because glucose is a primary source of energy for its normal function. So whenever there are low glucose levels, our brain cells are majorly affected. Glycogen breakdown, which is also known as glycogenolysis, is the first-line mechanism preventing glucose levels falling below normal. However, glycogen reserves are quickly exhausted during fasting or prolonged exercise. Metabolism must therefore be able to compensate by producing glucose from other sources. Various types of molecules may be degraded to provide substrates for gluconeogenesis. These include protein, lipids, and carbohydrates. If we talk about proteins, muscle protein is degraded to form amino acids, some of which, glucogenic amino acids, participate in gluconeogenesis. In lipids, mobilized fat stores or ingested fats, triacylglycerols, are hydrolyzed, releasing glycerol and fatty acids. Glycerol enters gluconeogenesis. Lastly, carbohydrates. We said earlier that it is generation of glucose from non-carbohydrate sources, so glycolysis under anaerobic conditions ultimately generates lactate, which can be converted back to pyruvate by lactate dehydrogenase. Pyruvate, in turn, is a gluconeogenic substrate. So that was all about the introduction of gluconeogenesis. In the next section, let us discuss the main process. Stick around.